Hey, my name is Frank Bucholtz. I'm a group leader here at the Planck Institute for Molecular Cell Biology and Genetics. And our work is maintained. We have two aspects of our work. One is concerned about RNAi screening. The other one is concerned about developing site-specific recombinases for advanced genome engineering. My background is that I'm a biologist, a molecular biologist, and I studied in Göttingen biology. I went then on to do a PhD at the European Molecular Biology Laboratories in Heidelberg, and then did my postdoc in San Francisco at UCSF with Mike Bishop, working on animal models for leukemia. The Max Planck Institute here in Dresden tries to combine, let's say, at least two different aspects, which is developmental biologists and cell biologists, with an attempt basically to, I think what one could say is that their basic theme is to try to understand how cells form tissues. That's basically the overall goal of the Institute here in Dresden. So we work on two fields, basically. One field concerns doing RNAi screens, trying to identify novel functions of previously uncharacterized genes in biological processes. The other one deals with genomic engineering to try to develop technologies that allow sophisticated engineering of genomes using site-specific recombinases specifically.